found out she was positive for SMA type 1, and we both kind of just broke down. We found out about the clinical trial here at Nationwide Children's Hospital.
This is really the first time that we've been able to apply gene therapy to any neuromuscular disease.
Today was a very special day for Evelyn. She was given genetic therapy for her missing SMN gene.
Babies with SMA are missing a gene vital for development. Through a one-time injection, gene therapy replaces this missing gene. For Evelyn, this gene therapy worked.
We start seeing changes as early as 2 months after treatment.
She started to push, and she started to get more active, holding her head up. And a little after 3-4 months, she rolled over on her own.
Now 3-year-old Evelyn challenges Dr. Mendel to a dance-off at her annual follow-up appointment.
She comes back after 3 years, and she runs up to me and hugs me and says, "Dr. Mendel, I love you."
I forget that she has SMA. This is a healthy girl, and she does everything that a normal 3-year-old child would do.
Something like that has never been achieved before.
With our first daughter, it was just devastating to lose a child. You lose all dreams you had for their future. And now we can actually save for college.
So I think the images speak for themselves. This is really the success of the development of a therapy for SMA, spinal muscular atrophy type 1. It really is linked to the in-depth work that was done both by the French Muscular Dystrophy Association and by Généthon. There's a history really of the development of Zolgensma. The identification of the gene responsible for SMA type 1 really depends on the collection of DNA from the family. And the first DNA collection was collected by the AFM in 1987. The first maps of the human genome, which have been a landmark tool in order to accelerate the development, the identification of genes responsible for rare disease, were developed at Généthon between 92 and 96. In 95, the gene responsible for spinal muscular atrophy, SMN, was deciphered by Judith Melky at Hôpital Necker, funded by the AFM. And from 2005 to 2010, Martine Barkatz at Généthon has developed gene therapy approaches for SMA and has demonstrated that a vector called AAV9 carrying a gene called SMN could protect mice from the effect of spinal muscular atrophy, which is what you see here on the right-hand side of the slide. You see here in gray SMA mice, which have a life expectancy of less than 20 days. And once treated, you see these mice living as long as 300 days, which was a good and unexpected surprise. Based on these findings and on the patents, the product was then developed in the United States, first by a biotech called Avexis, which was then purchased by Novartis and led to the launch of the drug Zolgensma. So this is an example, and let me go into more details about what is Généthon today. So we are a research leader in genetics and gene therapy. When I talk to audiences specialized in gene therapy, I usually say that we're probably one of the oldest companies working in gene therapy in the room, which is most of the time the case. We started gene therapy. We were created in 1990 and started gene therapy in 1997 at a time where there weren't out there many groups or many companies that were interested by gene therapy. And we developed treatments for rare neuromuscular disease, for immune system diseases, for blood disorders, for liver disease, eye disorders, and I will illustrate some of that. We go from upstream research down to clinical validation. Very importantly, we were created by the French Muscular Dystrophy Association, and we are not for profit. You could compare us to a biotech company. At European standards, we would be a large biotech organization. At US standards, we would be a mid-sized biotech organization, but you could compare us to biotech organization, but we are not for profit. And whatever revenues we get, we re-inject into research. We have a diversified pipeline of products, which we combine with technology innovation, and you will see how important technology innovation is in the field of gene therapy. And we're located 30 kilometers south of Paris, Évry, in France. So key dates. We were created in 1990 by the AFM Telethon. We established the first high-resolution maps of the human genome between 92 and 96. We started gene therapy in 1997, and we even started biomanufacturing, which, as you will see, is a key element in getting those drugs to patients. I mean, you could, treating mice is, of course, a very interesting step, but if you don't get pharmaceutical manufacturing, you will never get these drugs to patients. And we started manufacturing in 2006, which was quite visionary at a time where really there was nothing to manufacture. But as you will see, this was I think, a visionary anticipation. I can say this very very quietly, since I was not with the company, I was not with the association at that time. So it's not, I'm not talking about myself, we're talking about vision here. So key figures: 180 scientists and experts among a total headcount of 220. There's 12 products in clinical trial, which stem out of our research, which probably makes us one of the most productive R&D group in terms of getting drugs to the clinic. There's 7 programs which are at preclinical stage and which should get into the clinic within the next 3 years. Zolgensma is a product on the market, as I just shown, which integrates funding patents from Généthon. There's a second product reaching the market for an optic neuropathy, which is Lumevoq, which we have contributed to the R&D and the manufacturing. And now the product is held by a company called GeneSight, and we have major licensing and partnership agreements with pharma and biotechs, and I'll give more details in a minute. This is another way of looking at our history. Creation of Généthon in 1990, deciphering of the map of the human genome between 1990 and 1996, initiation of gene therapy in 1997, initiation of biomanufacturing in 2006, and then our first clinical trials starting in 2010. And you see here the list of the clinical trials of products stemming out of our research. An organization such as ours cannot hold a pipeline of 12 products alone. This would be the pipeline of a mid-sized pharma organization. We're too small, we don't have the resources. However, we are sponsors of 4 of these clinical trials, which are here in orange. The clinical trial for an immune deficiency, which is Wiskott-Aldrich, chronic glomerulonephritis disease, which is also an immune deficiency, a liver disease, Crigler-Najjar, and very recently, a clinical trial that we started for Duchenne muscular dystrophy, and I will give you some details in a minute. This is another way of looking at our pipeline. So we have programs here for muscular diseases. Here's the list of the ongoing trials for muscular diseases. We have a product under approval for eye disorders, an ongoing trial for liver disease, a product under clinical trial for sickle cell anemia, and a number of products which are under clinical trial for immune system and blood disorders. In yellow, outline those products for which we sponsor the clinical trial. This is the way we are organized. I said you could compare us to a biotech organization. One peculiarity of our organization is that we have integrated an academic unit from INSERM, which is the UMR 951, called Integrare, headed by Anne Galli and Anna Bushbilo, who are present with me at the Gene Therapy Congress this week in Washington. It's a 90 FT academic unit, so quite a large unit to which we provide approximately 90% of its resources, meaning that in this academic unit, there's 15 scientists which are on the payroll of either INSERM or CNRS, and the rest of the scientists are on Généthon's payroll. And we provide the unit with all its with all the support, both in terms of consumables and of equipment. We also provide the scientists with core services and platform, that being imaging, animal bioexperimentation, DNA bank. A very important part of our work is also technology development and CMC. CMC is everything that relates to the product itself and to the way of manufacturing it. And I'll come back to that. We have a clinical development and regulatory affairs group. We have a program management group in charge of organizing these multiple projects in a transversal way. And then we have an IP and business development group. We are handling a patent portfolio of approximately 800 different patents, 80 different families representing 800 patents covering our IP. The Academic and Translational Research Group has 5 subgroups, one which is immunology and innovative therapies, headed by Anne Galli, neuromuscular disease and gene therapy, headed by Anna Bushbilo, progressive muscular dystrophy, headed by Isabelle Richard, gene editing laboratory, headed by Mario Amendola, immunology liver disease lab, headed by Giuseppe Ronziti. And I have to say that with these labs and these lab heads really have a unique status, something that you don't often see in INSERM units. They are all at the origin, except for Mario Amendola, the others are all at the origin of at least one product which is in the clinic. Giuseppe Ronziti is at the origin of one product, Cridernaja, which is currently in clinical trial. Isabelle Richard is at the origin of one product which is starting clinical trial, and 5 others which should enter clinical trial within the next 3 years. Anna Bushbilo is at the origin of one clinical trial, which I will tell you about, which is led here by a US company, and a second product that will enter clinical trial at the end of the year. Anne Galli is at the origin of 4 different products. And I have to say that scientists at Généthon have a unique opportunity of transforming the research they do into actual drugs. So basically their work is to obtain new proof of concept for candidate gene therapies, to develop therapeutic strategies, to understand the physiopathological principle of the disease, to understand the immunological aspects of gene therapy. In fact, what we do when we do gene therapy is we inject something that looks like a virus, and I'll tell you about that. And you heard about it in the small video. So understanding the immune response of the organism to what we inject is very important. And they also work on technological approaches in order to develop new ways of delivering the therapeutic genes. And it's 90 FTEs. Another important innovation group is the technology and CMC development group. Think of the fact that today, 1 dose of these gene therapy drugs can cost up to several hundreds of thousands of euros. I'm talking several hundreds of thousands of euros per dose, which is clearly a limiting factor bring these products to as many patients as possible. So clearly here, this group works at reducing the cost per dose, increasing the quality of the vector, improving the productivity of the process, improving everything which relates to QC. And this innovation in the field of bioprocess is really a transdisciplinary approach. Hence, the group is working both with industrial companies such as Sanofi, Sartorius, but also with engineering schools such as ESPCI, the INSTBB in Bordeaux, but also academic institutions in France such as CEA, INSERM, and CNRS. And finally, the core services and platform provide support to the innovation group with an imaging and cytometry platform, preclinical assessment platform, and a DNA bank. Now, we clearly work at a crossroads of both public institutions and private for-profit companies. We ourselves are a private not-for-profit, and through our network with the I would say some of the best institutions in the field of gene therapy and rare diseases, through these collaborations and leveraging on our internal science, we come up with drug candidates that we can out-license to drug companies. And we have, of course, Zolgensma with Novartis, myotubular myopathy product at Astellas, Lumevoq at GeneSight, a Pompe program at Spark, which is a subsidiary of Roche, another CGD program, which is at Orchard, the Duchenne muscular dystrophy program at Sarepta. We're doing also bioprocess development with Sanofi, and bioprocess development with one of our affiliates, which is Hyposcesi. This allows us to have a global footprint, which we could not have on our own with our internal resources, and we could not develop all of these products worldwide. This is the map of the clinical trials which are currently testing or have tested products which stem out of our research. We're not sponsor of all these clinical trials, but these products have been tested, I would say, in a very large number of clinical centers, both in the United States and in Europe. This is our scientific advisory board. I will not go into the details. Amongst these scientists, you see Alain Fischer, all French. There's not a single French citizen who ignores who Alain Fischer is now post-pandemic, since he was responsible for the scientific and medical strategy for to handle COVID at the national level. But before that, in fact, Alain Fischer was responsible for the first successful gene therapy trial that was done in 1999, funded by the AFM. So he was a gene therapy pioneer. And you see in the scientific advisory board, which is an international board, both US representative, Karsten Bowman from the NIH, Annemiek Asma-Rus from the Netherlands, Amit Natwani from the UK, Francesco Montoni from UCL, Hildegard Bühning from Hannover. Now, also to mention that what we're doing at Généthon, we do it within a large ecosystem that was developed by the French Muscular Dystrophy Association. This ecosystem goes beyond Généthon. It goes from it includes a yearly call for proposal. It includes a large-scale manufacturing center under GMP rules, GMP being good manufacturing practice. And this is very important in order to get whatever material we need for clinical trials. It includes a clinical trial platform in Paris, a CRO for biotherapy trials, a biotech that we recently created focusing on myopathies, which is called Atamio, and an investment fund, which is able to invest in spin-offs of our program. I'm not going to review all of those in detail, but let me just give you some details. The call for project. So this is an AFM call for projects that funds 250 projects per year in academic institutions. Could be in France, could be in Europe, it can be in the US. Just want to mention that this fund has started some programs that were embryonic programs when they were started and funded by the call for project, but which gave rise to now products on the market. Zynteglo is a product now for beta thalassemia and a blood disorder. And the initial clinical trial was funded by this call for project. Skyzona is a product for a rare CNS disorder, which is called adrenoleukin dystrophy. It was funded by the call for project. The first ever gene therapy trial that succeeded for a rare immune deficiency for bubble babies done by Anna Fischer and Marina Cavazzana-Calvo was funded by this call for project, as well as the Lumovoq, which is a product reaching the market for optic neuropathy. Let me give you some elements about the investment fund. The investment fund was created in 2013 by the AFM Telethon and the French investment bank BPI France. It has funded several companies, which you see here, 50 million euro funds. Now the fund has reached its limits of investment, and it will be a new fund will soon be created. And then I want to tell you about Hyposkesi, which is a manufacturing platform, a CDMO. So it is a service platform serving both Généthon, but also pharmaceutical companies around the world, both in Europe and in the United States. It was created in 2016 based on our assets in bioproduction. It's a 10,000 square meter. It will have 10,000 square meter by the end of 2022. It is the largest European platform for the manufacturing of gene therapy products, producing both AAV and lentivectors up to the 1,000-liter scale, 180 FTs. So this gives you a view of the ecosystem in which Généthon operates. Now, let me go into some more details from a scientific perspective. What are we doing? We are doing gene therapy. So the principles of gene therapy are quite simple. We're talking here about rare genetic diseases. The objective is to correct a mutation, which could be both a mutation, an inherited mutation, or mutation that has appeared spontaneously during embryogenesis. We want to bring a piece of DNA into the nucleus of the affected cell. As you know, DNA does not spontaneously enter cells. It has to be transported. It needs a vector. The vectors we will use are viral-based vectors. That is, we will use recombinant vectors in order to bring the nucleic acid into the nucleus. And to do this, doing this, we can have different forms of intervention. This can be gene addition. We add a wild-type copy of the gene into the nucleus. So the mutated copy stays in the cell, but the wild-type copy allows to compensate for the deficit due to the mutation. We can also insert the nucleic acid to regulate gene expression. We can also do gene editing, that is, correcting in situ the mutation using a CRISPR-Cas9 approach. Today, all of our existing clinical trials are based on gene addition. Of course, we have more upstream projects on the other aspects. There's two different approaches for gene therapy, either in vivo delivery, and that is, for in vivo delivery, you take the vector with the corrected gene and you inject it directly either into the organ, for instance, at the ocular level, or by systemic delivery, that is intravenously. And from there, from the circulation, the vector will reach the organ. And this is the approach which is developed for muscular dystrophy, for eye disorders, for liver disorders, for hemophilia, for Huntington's disease. The other approach on the right-hand side of the slide is ex vivo delivery. For ex vivo delivery, you will take cells from the patient, and here you will take hematopoietic stem cells. So cells that give rise to the hematopoietic lineage. And this is mostly interesting for immune deficiencies, for blood disorders, for some CNS disorders as well, because microglial cells, central microglial cells, have a hematopoietic lineage. This is also interesting for CAR T therapies. I'll give some more details. So you take the hematopoietic stem cells from the patients, you introduce the corrective gene into these cells in the lab, ex vivo, and you reinfuse the corrected cells in the organism. This is what has been done for the initial trial for bubble babies by Alain Fischer, and it's done for many rare diseases, but it's also done for CAR T therapies. In CAR T therapies, the objective here is to treat oncology disease, blood cancers, by not replacing a missing gene, but by stimulating lymphocytes against the tumor cells. So the idea here is to transfer to lymphocytes a receptor, a chimeric receptor that will recognize specific tumor antigens, and that upon recognition will trigger a cascade leading to the destruction of the tumor cells. So as you see here, the gene therapy approaches for rare disease leads to applications into frequent disorders. We use two different types of viruses. On the left-hand side, the Adeno-Associated Viruses, small viruses, particle size between 18 and 26 nanometers, packaging capacity, which is 4.5 kb, so quite small, but sufficient for a number of applications, non-integrating. Adenoviruses are interesting viruses. Their physiological role is unknown. They are not associated to any type of known disease, and they're quite easy to engineer, where they're less complex to engineer than other viruses, which makes their interest, and they have a large spectrum of cells they can transduce. On the right-hand side, lentiviruses, which Madame Guyader, you know quite well, derived from HIV, integrating viruses, which are in particularly used for ex vivo gene therapy on hematopoietic stem cells, larger viruses, enveloped viruses with packaging capacity of 8 kb. Let me give you some examples of the achievements and of our research programs. First, here is a program, an ex vivo gene therapy for a rare and severe immune deficiency, which is the Wiskott-Aldrich syndrome, linked to the X chromosome, a rare disease affecting 1 out of 250,000 people. The mutation affects the Wiskott-Aldrich, the WAS protein, and the mutation causes hemorrhage, severe repeated infection, eczema, autoimmune reaction that can lead to cancer. These kids These boys have a limited life expectancy, and the only available therapy is a bone marrow graft, which does require a compatible donor. We have led an international clinical trial. The principal investigator at Généthon being Angali, who has designed the vector. It was an international clinical trial done both in the US, in France, in the UK. The initial results, so the trial started in 2010. The initial successes were published in 2015. And the long-term follow-up was just published in last January with a long-term follow-up of an average of 7 to 8 years, showing stability of a therapeutic effect and a correction of all major symptoms. These patients live, although they do not have a totally corrected, for instance, platelet function, they have a restored immune system, a restored T-cell system, which is very important, meaning that these kids live today a normal life. A second disease which I want to tell you about is myotubular myopathy, affecting 1 out of 50,000 live-born males. This myotubular myopathy leads to general muscular weakness. The muscle does not degenerate, but you have a general weakness which affects both the limbs, but also the diaphragm, meaning that these kids need ventilation, which can be a 24 hours a day ventilation. 50% of the kids will die before the age of 2. It is due to mutations in the MTM1 gene, coding for an enzyme myotubularin. And basically, there is no treatment. There's only palliative treatment. And clearly the prognosis is really a short-term death for these kids. You see here at the histological level the different types of defects that you see in the MTM muscles, both inflammation, central nucleation, decrease in size of the fibers. We have been developing, and this is Anna Bushbelo at the lab, which has been developing an AV vector encoding the coding sequence of the MTM gene, and has demonstrated the effect of this gene both in mutant mice and in mutant dogs. It happens that there is a spontaneous species of dogs showing the MTM mutation. I will not go into the details of the effects in mice. Let me show you the first results that we have obtained in dogs. Here's an MTM dog. These dogs usually have to be, are so weak that they have to be euthanized around the age of 25 weeks. So they really are pets. And let me show you the effect on such dogs.
Come on. Ooh. Come on. Could you try one more time? Thank you. Come on. She got something.
Come on, sweetheart. Oh, girl, come here. Good girl. All right, now call her.
Come here. Come.
All right. Can I call her?
So we're talking here about a unique injection in those dogs. What's really spectacular here is that not only we have a correction, but we have a reversal of the phenotype. The animal is weak, as you've seen before injection. It's, you know, it cannot walk by itself. It cannot raise. And the injection reverses the phenotype, which is not usual. I mean, you could expect with gene therapy to block the evolution of a disease. Here, in addition, you reverse the phenotype, and there's explanations for that, but I really want to stress that. So, based on this, on these results, we struck a deal with Audentes Therapeutics, a biotech company that was started exclusively around this program. So we licensed this program to this West Coast company, which was later purchased by the Japanese pharmaceutical group Astellas. The clinical trial started in September 2017. Now, this clinical trial is ongoing. I'll show you a video in French, but I guess most of the people here speak French. I'll show you a video about the unique French patient that was treated for myotubular myopathy, and then we'll come to the global results of the clinical trial. En plus, on n'entend pas. Let me check. Okay, so can you hear me? Pour ceux qui m'entendent, okay. Vous pensez qu'ils entendent quelque chose? Non, ben, so I'll make the comments. So
ça paraît tout petit par rapport à ce que ça peut apporter derrière. C'est une seule injection qu'il a reçue en intraveineux. Donc, il est resté quelques jours en observation à l'hôpital et on a pu ensuite le ramener à la maison. On ne s'attendait pas à ce que ça fonctionne aussi vite et aussi fort, c'est-à-dire qu'il a fait énormément de progrès en très très peu de temps. Avant le traitement, il ne pouvait pas tenir assis, donc il était allongé sur un canapé ou sur un tapis à longueur de journée. En termes de jeu, on était très limité parce qu'il n'avait pas de force dans ses bras, donc rien que soulever un hochet, c'était compliqué. C'était des aspirations régulières, puisque comme il déglutissait pas sa salive, il fallait bien le faire pour lui. Donc c'était ça le jour, la nuit, voilà, 24h/24. Jules, aujourd'hui, on ne l'aspire plus du tout, c'est-à-dire qu'il a acquis la déglutition, il gère sa salive tout seul. Il arrive à s'alimenter par voie orale. Qu'est-ce qui a changé? On n'a plus de ventilation mécanique la journée et on a eu le feu vert pour arrêter la ventilation mécanique la nuit aussi.
Allez, encore. Les mains en l'air, Jules, lève les mains. Lève les mains, allez. Ah, bien.
En fait, avant l'injection, Jules, il savait pas qu'il avait des jambes. Ça n'existait pas pour lui. Je pense qu'il avait pas, il réalisait pas en fait qu'il avait une partie inférieure au niveau de son corps, ses jambes, tout ça, c'était comme mort en fait. Les progrès qu'il fait, on a l'impression en fait qu'il les vit à 100 à l'heure, c'est-à-dire que avant il était comme enfermé dans son corps, prisonnier, et là il est passé d'un état neutre à un état complètement euphorique. C'est plus, c'est-à-dire que tout ce qu'il vit, il le vit comme si c'était la première fois, enfin, mais c'est sa première fois en même temps. C'est une deuxième naissance, c'est-à-dire que Jules, il a 2 anniversaires. Il a le 3 avril 2016 quand il est né, et il a le 29 janvier 2020 quand il est rené. C'est-à-dire que quand il a eu sa deuxième vie, sa deuxième chance, et je pense que nous, on va fêter cet anniversaire-là comme on fêtera son anniversaire de ses 4, 5, 6 ans.
Allez, vas-y. Voilà, c'est bien. Pousse. Ouais, super. Et on s'assoit. Voilà, c'est trop bien. Stop. Super.
So the clinical trial has recruited 24 patients. What are the main results? So On the motor function part of it, several of the patients have reached ability to walk unsupported. In the treated groups, there were 2 cohorts at 2 different doses, while none of the untreated patients reached the ability, could ever walk. On the respiratory side, several of the kids reach ventilator independence, whereas a boy like Jules, for instance, needed 24 hours ventilator support, and none of the control group did reach ventilator independence. However, there were severe adverse events in 4 of the kids, leading to liver dysfunctions, and 4 patients disease from liver dysfunction, meaning that this trial is now on hold. The reason of these deaths are unknown, and probably this disorder, multiple neuropathy, is not only a muscular disorder, but it could also be, I mean, could also be a liver deficiency. And it is true that the quantity of product that is injected is high, and could be stressful for the liver. And in kids which have a liver deficiency, this could lead to severe adverse events. So these adverse events are under review with the objective of restarting the trial, because you understand that the families are pushing really hard. Whatever the risk-benefit ratio, I mean, the risk-benefit ratio is really in favor of the benefit. I mean, the parents say, well, you know, we don't care about the side effects. We know exactly what will happen to our kids if he's not treated. So there's also an ethical part to that, and a regulatory part to that, because of course the regulatory agencies and pharmaceutical companies don't want to progress a compound, a product, if there's a lethal risk attached to it. Now, another program I will rapidly, for the sake of time. How much more time do I have, Madame Guédère? It's already done. Okay. Oh, give me 10 minutes. Okay, thank you very much. So another program I would want to stress is Crigler-Najjar syndrome. Crigler-Najjar syndrome is an ultra-rare disease affecting 1 person out of 1 million. So this is a deficiency of a liver enzyme, which is responsible for the elimination of bilirubin. Bilirubin is a yellow pigment, which is formed from the recycling of blood cells. So the accumulation of bilirubin causes the yellow color, which you see in jaundice, and it could be and just this color is not, I would say, a severe pathology by itself. Some kids are born with a jaundice, and they spend 2 hours below a blue light, and it's done. Here in these kids, they never eliminate bilirubin, and if bilirubin is accumulated in the body beyond a certain threshold, it will penetrate the central nervous system and cause irreversible neurotoxic effects. So clearly, these kids, and then when they become adults, they have to live under a blue light at least up to 10 to 14 hours a day, which is the only treatment. So you can imagine, I mean, it's simple enough to have a newborn under blue light, but as the kid grows and as he becomes an adult, this is the kind of setting he has to undergo every day for up to 14 hours a day. We have developed a gene therapy, an AAV vector encoding the normal version of the gene encoding the UGT1A1 enzyme, which is targeted to the liver in order to express the normal protein and lead to the elimination of accumulated bilirubin. The product was first demonstrated, was first tested in rats and mice affected with the disease, transgenic animals. And you see here total bilirubin at different doses. So the bilirubin levels under different doses of treatment with the AAV vector. You see here increasing doses of the vector, here in dotted line a low dose, here in full line a higher dose of the vector, showing a decrease of total bilirubin down to 0 in the treated animals. You see here on the right-hand side the color of the serum from treated or untreated animals. A clinical trial was started with this product. I will not go into the details, but the principle is the following. It is to inject the product to the patient, to wait, to look at the decrease of bilirubin, to wait for a latency period, and then stop phototherapy. What you see here in these 3 patients is that upon injection of the product, you see decrease in bilirubin, and this decrease was deemed long enough so that phototherapy could be stopped. And you see here in this first patient, phototherapy that has been stopped for several months, several weeks for this patient here, and this one and this one will probably stop phototherapy also. So in fact, I mean, we have more recent data than that. And this patient has been off phototherapy for almost a year, for more than a year now. For the first time of her life, this patient has not needed phototherapy and has led a normal life. And you see here, it's visible from the color of the skin, but it's not the most important element. It's really the quality of life which is impacted. So this clinical trial is ongoing, and we'll see how long is the effect and how we can have a prolonged an effect that is as prolonged as possible. I want to spend a few minutes on Duchenne muscular dystrophy. I started my presentation with the fact that we were created by the French Muscular Dystrophy Association, which was created around Duchenne muscular dystrophy, which is, I told you about this disease, a progressive and a fatal disease, which affects 20,000 patients in the United States and in Europe, 200,000 patients globally. It is due to a mutation in a gene which is the largest gene in the human genome, which is the gene encoding dystrophin, which is a very important structure, which encodes for an important structural protein in the muscle, which really makes the link between intracellular actin and the extracellular matrix. And mutations in dystrophin will create a weakness in the muscle, leading to progressive microlesions. An accumulation of microlesion will cause a degeneration, irreversible degeneration of the muscle. This gene is too long to fit into an AAV vector. And with George Dixon at Royal Holloway, University of London, we have worked on reduced dystrophin, what we call microdystrophin, which recapitulates the major functions of the total dystrophin, and that can fit into an AAV vector. I will cut a long story short and show you the effect of these vectors in GRMD dogs, which are dogs which show mutations in their dystrophin gene. So this is an affected dog, untreated dog. This is a treated dog. So 2 dogs, 1 untreated, 1 treated. Two treated dogs. A 19-month-old dog. That they rarely get to that age when they're not treated. Okay. Now, I will not be going into the details of the studies. We've been studying both the clinical score of the dogs, and you see here in blue the treated dogs, in red the untreated dogs. Clearly, a statistically significant improvement in the clinical score of these dogs. We've been studying the gait of these dogs. Although they have not recovered once treated, they have not totally recovered once treated. They're strongly distinct from the untreated dogs, which are here in red. So these are in yellow, the treated dogs, in green, the normal dogs. I will not go into the details, just for you to know that one of these dogs, born in 2013, treated in 2014, is still alive. So he has been kept to see I would say the longevity of the effect, and this has never been seen. Never seen a GRMD dog living 8 years after treatment. And basically, there is a clinical trial ongoing with this product, GNT004, which is a clinical trial approved in France, in the UK, which will submit also to the FDA. It's an ongoing clinical trial. Too early to talk about the results, but this is ongoing. An important turning point both for Généthon and for our parent organization, the AFM. Now let me conclude with the challenges that we have ahead for gene therapy and that we're addressing at Généthon. One thing is that we have to, I didn't touch in too much detail at this point, but we have to better understand and control the immune reaction to in vivo approaches. What we inject is something that really looks like a virus. I mean, to the organism, this is a virus. And this has several consequences. There is the activation of an immune response, both innate, adaptive, cellular immune response, which has several consequences. First major consequence is that you cannot re-inject. You cannot redose the patients. Once the patient has been injected, it cannot receive the product a second time because he has both antibodies, memory T cells, and a second injection will not be efficacious. And this also clearly can be a limitation. Why can it be a limitation? You've seen some very long-term effects in the programs I've shown you. But who knows what will happen in 5 years, 10 years from now? In kids who are growing, the so-called concentration of the therapeutic transgene might drop below a therapeutic threshold. So you might need to re-inject. If you lose the effect, you might need to re-inject. So controlling this immune reaction, being able to temper it, is an important element in the fact of redosing. Also, in clinical trials, think about the fact that clinical trials are built upon an increase of dose, a dose escalation. You treat a first group of patients at a lower dose, and then you increase the dose up to the moment where you find the appropriate dose to get the clinical effect. But those patients who will have been treated with lower doses, basically, you will not be able to treat them again. So they've been treated in a clinical trial, but they will not, if the dose they received is not efficacious, you will not be able to treat them with the efficacious dose. And then the third point is that the immune response can be at the origin of a toxic side effect. So clearly, better understanding the immune response is an important element, and we are developing programs at Généthon in order to better understand this immune response and better controlling it. We have to reduce doses, improve product efficiency, so have products that can be injected at lower doses, both because you always prefer injecting lower doses than higher doses whenever you inject a product. Just to give you an idea of doses, these are recombinant viruses. Gene therapy is not the only area where you use recombinant viruses. In fact, you use recombinant viruses for vaccination also. Think about the AstraZeneca vaccine for COVID-19. This is a vaccine which is based on a virus, which is adenovirus, not AAV, but adenovirus, transporting the gene for the spike protein. When you inject the adenovirus to elicit an immune response, you inject 10 to the 10 particle per dose. For Duchenne muscular dystrophy, you inject 10 to the 16 particle per dose. So we're talking here about much higher doses, which raises questions both in terms of what type of side effects you might trigger, but also in terms of production, cost of production. I told you about the production, which had a cost of approximately several hundreds of thousands of euros or of dollars per patient. So clearly reducing the injected dose, improving the efficiency of the virus is something important. Decreasing the cost of manufacturing, same element. You have to be decreasing the cost of manufacturing if you want to be able to have these drugs made available to as many patients as possible. And the cost of manufacturing is important. Thus, the price of these drugs and the pharmacoeconomics of these drugs is specific. The most expensive drug today is a gene therapy treatment. It's a gene therapy treatment for rare CNS disease, and it's priced 3.4 million pounds per treatment. Think about that. Zolgensma for the spinal muscular atrophy is priced at approximately $2 million per treatment. So clearly, decreasing the cost of manufacturing, which is a part of the price, is important in order to get better access. Again, on this aspect of pricing, manufacturing is not the whole element of the price. I mean, the pricing aspect, there's also the pricing strategies from the pharma companies. Pharma companies, based on the fact that this is a unique injection, argue that they have to recoup their investment, their R&D investment on one unique injection as compared to chronic treatment, and thus claim very high prices. We think, as a patient, as an R&D center created by a patient organization, that this should not be the case, that these prices are too high, and they are a limit to the access to patients. So clearly, working on pricing is an important element. And then there are these rare diseases which have no commercial model, because there are some rare diseases which affect only 50 patients per year, both US and Europe. And whatever the price of these the treatment can be, there will be no return on investment, because development is expensive and development is costly. It's several tens of millions per treatment, whatever the number of patients is. But you've seen the efficacy of these approaches. And if we don't find a way of financing research and development, even for these ultra-rare disease, there will be multiple patients that will be left on the side of the road. So clearly we have to find here creative consortiums, public, not-for-profit, for-profit partnerships to find the ways of financing this R&D. In the US, there's such a consortium that was started by the FDA and the NIH, which is called the Bespoke Gene Therapy Consortium, which regroups both, which was created at the initiative of the NIH Foundation with the FDA, which groups several pharma companies, for-profit companies, not-for-profit. Généthon belongs to this consortium. It's the only non-European institution that belongs to this consortium. And here, what we would like to do is, based on what we learn in this consortium, also start a comparable initiative in Europe. I could be speaking about what we do about gene therapy for the rest of the night. This is not my objective. However, you don't want to spend your evening here. And I thank you for your attention. If there's any question, I'll be happy to answer. Thank you. I will stop sharing.
We can have questions from the virtual participants too.
So you were mentioning that you focus mainly on Europe and the US. Are you considering to look also more into other parts? I mean, rare diseases are everywhere around the world. Are you interested also to collaborate with institutes in Africa or in Asia, or expand that too?
Yes, we definitely are. So I think we have been collaborating at one stage with Brazilian institutions. I think the specific skills which are needed to treat patients with gene therapy are quite specific, probably quite expensive. And we've seen some South American groups interested in testing the product. It never we never made it at the end. I mean, we had many discussions, but it never came to a result. But I think that for some diseases, in particular of the African continent, gene therapy is an interesting approach related to compliance. We perfectly know that cost is an issue in Africa, and I think the day has not come where where many African countries can afford treatments at several $100,000 per patient. But nevertheless, we have to think about one thing. In countries which do not have coverage in hospital services, where compliance is an issue, having a once-off treatment can be an interesting solution. Think about sickle cell disorders. If you could finance a one-off treatment and not have to distribute treatments that would have to be injected on chronic basis, every week, every month, it would improve the compliance in these underdeveloped countries. So I think although the cost is important, in the countries that do not have the hospital infrastructure that would allow a complete coverage, gene therapy could be of interest. A little bit like a cell phone has developed in African countries without having regular phones or ground phones ever being developed there. So that's
one question in the chat.
So what is the question? Let me see that. So
my question was to
are you going to address some of the neurodevelopmental disorders than neuromuscular disease?
Okay. So, what do you mean by neurodevelopment disorders?
Like autistic, ASD, autistic syndrome disorder. So we know that there are a lot of disorders which are in the single gene mutation, and like I'm thinking of Angelman syndrome or Rett syndrome, something that we know the nature of a gene. And are you planning to address these?
Yeah. So we have right now no programs ourselves for Angelman or Rett's diseases. So that there's some neurodevelopment disorders that are based on dopamine deficits. And I'm sorry, because there are 7,000 of these diseases and I have the name on the tip of my tongue, but so we're addressing one of these. So there's some programs out there also in other institutions for some of these disorders. So we have to keep in mind that when we talk about neurodevelopment disorders, whatever is already damaged at birth might not be corrected. Okay, so there's always the idea of in utero gene therapy, but I think it will be sometimes before this approach is really developed, because it does really raise a question of biodistribution and also of the setting of the clinical trial, and at the end also the risk-benefit ratio between an abortion and an in utero treatment. So when we talk about neurodevelopmental diseases, we have to address those diseases for which there's no strong damages at birth, because whatever is damaged at birth will not be reversed, will probably not be reversed. Do we have?
Thank you. I have one question. Are you looking for, do you have any opportunities for jobs or
yeah,
young scientists?
So we are actively recruiting in different fields. So it's both in the development of novel therapeutic entities, working in so we both welcome scientists which would join an existing group developing therapies, neuromuscular diseases, liver disorders, but also we have recently recruited a scientist coming with his own program and his own project for a given disorder. So that's one element. We are recruiting scientists in the field of immune response to AAVs. We are recruiting scientists in the field of improvement of bioprocess. And improvement of bioprocess is really at the crossroads of different disciplines, means in understanding the way a virus is assembled in the cell. It means understanding what happens in a bioreactor. It means improving analytical methods. So, and we're recruiting at different levels. So we're recruiting at the postdoc levels, temporary positions. And I have to say that right now we have more postdoc positions open than we can fill. I mean, it is quite difficult, and I think it's not only a question for us, but for several institutions. France are recruiting interesting postdocs. Maybe it was more difficult than before the COVID crisis, but also we're recruiting permanent positions. So being I would say a mid-sized organization, we're also quite flexible in adapting to so we're private, so our decisions we make ourselves. Okay.
So you have like package kind of thing.
So depending. So we have so of course we have the salary. We put in place relocation packages for scientists coming from the United States, helping them relocate in France, helping them move to France. And depending on the seniority, it could be also providing support, technical support, technicians, creating a group. So that's what we do, and basically, yes, that's we have at different levels. But I want to stress that we are being private. Our mode of recruitment we do ourselves. So that, I mean, we're not the INSERM, we're not the CNRS, so we don't have these commissions. Clearly, we recruit scientists based on their dossier, based on the fact that they would meet the senior scientist and we would make collegial decisions. Yeah.
And so if you have people interested, they can contact you directly, or do you have any contact?
They would contact me directly, or the HR department at Généthon. They contact me.
More question? No?
You actually asked the question. Do you have like any website just to look at the?
Oh, we have a website. We have a. Yes, you can apply online on our website. Our website is open for online submission. Yeah. In French and in English.
Okay. I guess we will, thank you.
Thank you very much.
Thank you for the one who came here at the embassy, and thank you to all for listening online to Dr. Revah. If you have any further question, we would be happy to answer them. Don't hesitate to contact us, and we will forward your question to Dr. Revah. Thank you to all, and have a nice evening.